Okay, let's get started in class. So in the last lecture, we started explaining Snips, linkage disequilibrium, and GWAS studies. And we also briefly talk about EQTL. And so today we're going to talk about EWAS, which is how to use epigenetics to understand a GWAS study. So as we mentioned, the number of GWAS studies has increased drastically over the years, and there are many GWAS Snips that has been identified to be associated with the disease. And if you look at the total number of, well, this is a 2012 paper, you can imagine this continue to grow. On the left side, this explains the number of GWAS studies that are associated with different type of phenotypes or diseases. Whereas the right is, if we look at all those GWAS hits, where they are. Remember, initially people were hoping that, oh, we find this is on a gene, we will know this gene is related to the disease, but to people's surprise, most of the GWAS hits actually don't land in genes. So this is only less than 5% land in the coding regions of genes. Small percentage, 1.4% land in promoters of genes, but most of these are either in introns, like 40% of those GWAS hits are in the introns, as well as regions that are either within 50 kb, 50 to 100 kb, or even 100 to a megabase away from some genes. So a lot of these GWAS hits actually land in very gene desert regions. And so it's a very difficult question about what those GWAS do, which ones are real, what genes do they regulate? And to understand this, so in the early days, people who are doing GWAS studies are mostly epidemiologists. They have some big cohorts, they are studying the disease for many years, they have blood or some maybe tissue samples for some individuals, or big cohorts of case controls. And they do either SNP arrays or whole gene or exome sequencing, or nowadays whole genome sequencing. But once you have identified all these GWAS SNPs, in order to really understand which one is real, why these SNPs are important, now people are looking very heavily into epigenetics and transcription regulation. In the previous lecture, we mentioned the EQTL. Basically, instead of using the disease phenotype or a trait like a height or body mass index as phenotype, we can use the expression level as a phenotype. Right? We want to ask whether the presence of some SNPs is associated with expression changes, because there are sometimes heritable genetic variations for the transcription levels. And based on the distribution of all the GWAS hits, people are suspecting that a lot of these trait or phenotype associated SNPs are RSNPs or regulatory SNPs, which means that they land in a region which is potentially bound by a transcription factor. And then that transcription factor might regulate a nearby gene. Yes, Jet?
When they do EQTL, do they look at overall expression, or do they look at how one SNP changes the expression of every other gene in the genome, or is it just genes nearby?
Okay, that's a very good question. So Jet mentioned this question about whether they look at the genes that are close by or far away, right? So first of all, in terms of gene expression, as we know, different parts of our body have different gene expression. So you might want to do this tissue by tissue. Previously there were mouse studies, but now there are also GTACs where people they just donated the cadavers and they take out the different parts, like different tissues, different organs, they do expression, right? And they also have the genotype of those individuals. But in terms of the EQTL, you can imagine, you can look at the correlation of a SNP with a nearby gene expression, say within a few hundred kilobase or within a megabase. And these are usually called the cis EQTL. Cis just means it's on the same chromosome, but usually it means it's pretty close to the SNPs you are looking at. Yeah, most people look at it within 1 megabase of the SNP. But there are also cases when there is a trans EQTL, which means that the SNP is on one chromosome, but then when you look at the expression of other genes, they could be on other chromosomes. But you can imagine if we're going to do this, the number of tests will increase a lot. The multiple hypothesis problem will be much bigger. And so you need to have a very, very stringent p-value, or at least adjust for false discovery rate to identify those. Okay? Yeah, so let's go back to this. So because most of the GWAS SNPs, those hits turn out to be in non-coding regions, people start to suspect that maybe they are regulatory SNPs, which means that these SNPs might be bound by a transcription factor, and because of that SNP, maybe the transcription factor can bind better or worse. Maybe that SNP disrupts the motif of the binding. So the transcription factor can bind at different levels, and depends on that binding, it might influence the expression of a nearby gene. Yeah, so you can see here in human, mouse, and mice, 30 to 50% of the genetic basis of intraspecies differences in the transcription level are cis to the coding locus. So even though the SNP itself is non-coding, they are related to the nearby gene. And so in the same study that look into the number of GWAS SNPs, they look at all the GWAS SNPs. And so you can see here, we mentioned that there is an experiment called DNase hypersensitivity sequencing, or DNA sequencing, which basically give us all the binding site of a transcription of different transcription factors in a particular cell state. You basically get all the collection of binding sites, and the group that is who is specialized in this technology, they also have done DNAs in many, many different cell types, like in hundreds of cell types. And the union of all of those DNA sites pretty much give us a quite complete set of all the potential regulatory sequences in the genome. And when you overlap all the GWAS SNPs with the union of all the DNase hypersensitivity sites, you can see here, they at least in this study, they found 57% of the time, the GWAS SNP land right in the DNase peak, which means that very likely that particular GWAS SNP will change transcription factor binding. And then there are another about 20% of the cases where the GWAS SNP is in perfect LD with another SNP within a DNase peak. Right? And then another 23% is the case is the GWAS SNP is not in LD with the, so those ones, I guess, maybe DNase hasn't profiled those cell types yet, right? It could still be a transcription factor binding site. And so those we don't know for sure, but it would mean probably 3/4 of those GWAS SNPs might be related to transcription regulation and transcription factor binding site. Yes, Sam?
For the SNPs that are kind of in those cancer regions, 25%, do they have a smaller effect size than the other SNPs?
So the question is, for those GWAS SNPs that land in gene desert, whether they have smaller effect size. I actually don't know the answer to that question. My suspicion is that a lot of people are very interested in those because they do show, my suspicion is that they actually are pretty strong. If it's in a gene desert and the effect size is small, p-value is not so significant, people wouldn't go there anymore, right? The reason people are looking so hard is because there is a very strong GWAS and it could be associated with multiple disease. And there's not a gene nearby, and people are wondering, what is this SNP doing? Right? So that's the big question here. Yeah, and so this is in figure A, and in figure B, this part is also quite interesting. They are looking at the percentage of GWAS SNPs that are in the DNase hypersensitivity regions. And what they found is that if this GWAS SNP land in a DNase hypersensitivity region, which means it's probably bound by a transcription factor, about 70% of the chance it will be externally replicated, which means in different studies, they actually can replicate each other. Yeah, and then, but whereas those unreplicated SNPs, they are less likely to reside in the SNP. So the real ones are probably going to be related to transcription factor binding. And because of all these findings, people now are looking very heavily into ChIP-seq, looking at the transcription factor, ChIP-seq, histomark ChIP-seq, and the DNA's hypersensitivity regions now. Yeah, so in order to understand this, sometimes people use this. For example, if you look at the histomark or DNA's hypersensitivity, sometimes people can also use the histomark ChIP-seq signal as a quantitative trait instead of gene expression. So ideally, you want a SNP to directly to be associated with a phenotype like cancer or some disease. EQTL is trying to associate this with expression, but sometimes we may not even have the expression. We can just, so in order to do EQTL, well, in order to do GWAS studies, you usually need like hundreds or thousands or tens of thousands of people to do an experiment. In order to do EQTL, at least you need probably like 100 like different mouse or mice to do this experiment. But recently people are looking at histone modification or DNAs, and there they might only need a few samples to look at this, or even a single sample to look at this. For example, supposedly this is the ChIP-seq of one individual. You might, or a few individuals, you might look at their ChIP-seq or DNA signal for those individuals at this locus. If they have the AA allele, the genotype, you can see their ChIP-seq signal around here is pretty high, but if they have AG, which is a heterozygous, you know, the signal is weaker, whereas if they have the GG signal, then the GG genotype, then the signal is pretty weak. This would suggest that maybe there is another SNP in LD. So by the way, this is H3K4 trimethylation signal right at the transcription start site, or at the transcription start site of some gene here. And so there could be another SNP in LD that's nearby with probably like a taxic signal or DNA signal or H3K4 monomethylation signal in there, which is in LD with this particular SNP. And potentially that SNP is changing the transcription factor binding, which will also change the promoter activity of this. So you can see here, basically, if this region have the A allele, the promoter will be probably more open, the gene will more likely to be activated more. Whereas if it has the G allele, the promoter will have lower signal, probably less gene activation. But if it's a heterozygous between A and G, then it's intermediate signals. And so here you can see, even from a very few number of samples, you can already gain some insight on what's going on with this particular locus, right? And so people have, yeah, so this is a promoter signal. As we mentioned, very often the promoter signal might be also related to an enhancer signal. This is another EQTL in the sense that the E is not expression, but epigenetics. For example, these are different ratio groups. And you can see because of the ratio groups are different, they might have the GGAA, or in this case, CCAA allele, right? And you can see here, if they, so if it's a one ratio group, in this region, this DNA's peak is present. And this is the, I think, yeah, so you can see here, they have the, if they have the GG allele, the signal is very high, but if they have the AA allele, the signal is weaker in this location. And you can see also different races. In this, there's a peak in one racial group, but no peak in another group. Whereas in a different region, there might be a big peak in one group, but no peak in the other. And you can look at their genotype. You can say, oh, turns out because of their genetic, in that region, the SNP is different. There's a different peak. And then you can try to see whether that DNase peak later on regulates the nearby gene expression. So, so far, unfortunately, for this particular study, this is done once you do racial groups. You can't take out their tissues. You can only draw their blood. So you may or may not have enough signals, right? For example, if this is a neuronal disease or it's related to liver, you may or may not be able to see it. Ideally, you want to do epigenetic profile of different organ or tissues as well to see whether that particular SNP is associated with a differential peak in those different racial or SNP individuals. Okay, yeah, so you can see here, very, very often it's because, yeah, so in this case, the reason that this binding site or this region has a differential DNase peak is because there is a CTCF transcription factor that can bind to here when the sequence is 1. But if you just change a single nucleotide, the same CTCF cannot bind to this region anymore. And this is vice versa in a different situation. Because of the SNP, the CTCF is only binding in one racial group, but not binding in the other racial groups. Okay, yeah, so those can have big effect on the nearby gene expression. So this is related to one of Sam's question. So there is one GWAS SNP, it's RS9832667. It's on chromosome Q arm, so the chromosome 8, Q arm 24. And this region, it's interesting. Initially, people found to be associated in colon cancer, but later on, multiple GWAS studies have identified this SNP as associated with multiple cancers. And yet, this region is a kind of a pretty sparse gene desert. There's not much going on. And so basically, so in order to find out what this SNP is doing, this particular group just look at the histone modification data and they ask what is in here. And indeed they found, oh, there is a ChIP-seq peak in this particular SNP location. And they ask, okay, then what are the nearby genes? And when they keep looking further and further, they found, oh, the nearby gene might be MYC. Okay, and so in order to really show that this is related to MYC, this group did a chromatin conformation capture experiment. Do you guys remember the Hi-C experiment that we discussed in the lecture last week, or sorry, right before spring break? So Hi-C is looking at all genome-wide pairwise interactions, but 3C chromatin conformation capture is just measuring how much interaction do we see from one point to another point. So they tried all, like from one, this particular location to the beginning of the MYC gene, from 2 to the beginning of the MYC gene, from 3 to the beginning of the MYC gene, and see how much interaction they are seeing. And they can really see that this particular SNP, even though it's really, really far away from the beginning of the MYC gene, the enhancer is looping to the promoter of the MYC gene, which suggests that that particular SNP that's very, very far away might be bound by a transcription factor which regulates the expression of MYC. And MYC is a very, very well-known oncogene. When its expression is high, people are more likely to get cancer. And what they found is that this SNP is bound by a transcription factor TCF7L2. And also, again, whether the SNP is one or the other, it changes the binding strength of the transcription factor, which then in turn regulates whether the MYC expression will be high or low. Okay. This is another study. So there are many, many studies now recently looking into this. This again is a Snips study related to prostate cancer. And this particular SNP, also you can do this overlap with either ChIP-seq data of histone mark or ChIP-seq data of different transcription factor binding sites. And so in this case, it's a ChIP-seq of some transcription factors. It's HOXB13. And when the allele or the genotype is CC, you can see the peak is very weak. If it's a TC, then the peak is stronger, but if it's TT, HOXB13 combined much stronger. You can even do a ChIP-seq on the patient tumors to see whether the CC, TC, or TT looks different. And then they just look at the nearby gene. Turns out that the nearest gene to this particular binding site is not even active in that cell type. So they just kept looking further. And then there is a nearby gene, and they did find that when this particular SNP is CC, the nearby, or it's not nearby, it's like pretty far away, but the RFX6 gene expression is lower, but when the peak is strong, the RFX6 gene is much higher, right? And then they look at the public data to ask, is this RFX6 gene related to prostate cancer? Indeed, they found that in 2 different prostate cancer cohort, when the RFX6 gene is highly expressed, you can see here, the red is high gene expression, blue is low gene expression. Oh, by the way, we haven't discussed this yet. It will be in the next module of cancer genomics. This is a curve called the Kaplan-Meier curve. It's just asking if we were to separate the patient in 2 groups, in this case by the expression level of this RFX6 gene. So we evenly divide the patient into RFX high or RFX expression low group, and we ask from the time of diagnosis, if we wait for many months afterwards, whether the patient will still be alive, how many % of the patients are still alive? And you can see here, basically, if those patients have high RFX6 gene expression, they have a higher chance of dying. So they have a worse clinical outcome. Okay, so that kind of explains this particular SNP. Not only explain, like the binding of some transcription factors, it explained that it influenced the expression of a nearby gene, and that gene is very important for probably prostate cancer development and also progression, and eventually might lead to the patient outcome. Okay, yeah. So in general, one thing that people discovered also was kind of interesting. The highest correlated genes of distal DNA's hypersensitivity region compared, and when they were able to link to a nearby gene, you can see sometimes there's like a very good correlation. This is correlation of the DNAs from the transcription factor binding site, so distal sites, to the DNA site in the transcription start site. They look at the signal correlation between the enhancer and the promoter. And so you can see here for one disease, this particular SNP and the disease gene is having a correlation of 1. But when they look at the distance, they were very surprised. The distance is very long. It's like 400 kilobase away. Most of the time, a transcription factor would only regulate genes that are, maybe 10 kb or 50 kb away. But a lot of these GWAS regions, when they look at the disease SNP to the linked disease gene, and then when they try to look at the distance, you can see this one is 20 kb, this one is 100 kb, but many of linked genes are like 100 kb or even longer, which means a lot of these GWAS SNPs are in kind of a gene desert region. There are usually not so many genes. When they have a GWAS SNP, they just keep looking, because either there's no genes there or the nearby gene is not active, so they keep looking until they find the gene that's, it's active in those kind of disease settings, or at least the right tissue, and also is active. Okay, so yeah, so right now, in order to link the GWAS SNP to the nearby gene, it's still a very big scientific question. People are trying to look at signal correlations between the promoter and enhancer in terms of its DNA's hypersensitivity signal, in terms of its like ChIP-seq signals, or using the high-C data to see whether there are chromatin interactions between that SNP and the nearby gene. And very often they would find that the target genes are pretty far away. Okay, you can also just use ChIP-seq data. This is related to homework 4. Based on all the binding for this transcription factor, figure out what its putative target gene is going to be. Next, this is related to Jet's question. Out of all the SNPs, in early days, most people are interested in cis SNPs, which means there is a SNP here which is related to the binding of one transcription factor, and that influences the gene nearby. Nearby, we're talking about like 50 kb or 200 kb, but at least on the same chromosome. But there are trans EQTLs where the SNP of one one SNP somehow influence the expression of many genes on different chromosomes. Can you guess what is happening there? Some students suggest super enhancer. So the super enhancer might have an effect, but a lot of super enhancer target genes are transcription factors too. So you can imagine if there is a SNP, it might influence the binding of a transcription factor, and that could influence another cis gene, which is a transcription factor, right? Then when that transcription factor has differential expression, then it will bind differently to many of its target genes. Then it could have a much, much stronger downstream effect than just the SNP itself, okay? And so, if you look at, so people started looking at these trans EQTLs, and they found, for example, in this region, so each of this is a different block in the genome, and you can see this region, by the way, each dot, each little, this circle is one trait so you can see here, many studies are finding this, or many SNPs are within this region, and somehow it's related to EQTLs of faraway things. And so it turns out this region has a very, very important transcription factor. And if you have anything near it that changes the expression of that transcription factor, it's going to have a very strong effect of many downstream genes. Yes, Jet?
When they look at like trans EQTL, do they compare like how one SNP affects every other gene in the genome? It seems like that would be a super large number of tests.
Yes. Yeah, that's what they are doing. It's hard. And so then only the very strong signals show up, right? And then this is kind of from many tissues, from many different studies. You might see this. Okay, then that's probably real. You want to find out what's going on with these. Okay, yeah. So this is one study that was published in Cell. It's kind of interesting. So they look at some risk loci that are near transcription factors. So this is one these are all actually cancer-related risk loci. And one is near the estrogen receptor gene, one is near the MYC gene, and one is near the KLF4 gene. Turns out all three are transcription factors. And just based on the GWAS studies, if you try to look at whether the SNPs in the enhancer region change the transcription factor expression in a cis EQTL fashion, the trend is not very strong. So maybe there is gene expression changes, but it's very, very small because transcription factors are not usually expressed at very, very high levels. And so when the expression level is lower, it might be pretty noisy. You may not see a very strong signal. However, because these three are all transcription factors, they can look at the ChIP-seq data of these transcription factors and look at their putative downstream target genes. Okay, so you can see, and then they look at all the downstream, it's like a gene site enrichment analysis. They're asking, when you have this particular one SNP or the other SNP, are the downstream of estrogen receptor genes having a big difference between one allele or the other allele? And so instead of looking at estrogen receptor gene expression alone, they are looking at the estrogen receptor transcription factor targets. There you have many target genes, and then they actually have much, much better power to detect the difference. And those are a trans-EQTL effect. And so there, instead of looking at all the genes, they only look at the target genes of estrogen receptor or the target genes of MYC or target genes of KLF4. There they have much better power. Okay, then yeah, you can reduce the number of tests. So all of this is, I would say, a lot of research in progress. So people who are interested in precision medicine, you know, like right now there is a precision medicine initiative at NIH. They just profile like 2 million healthy individuals. Some have disease, some have just healthy individuals, but you have different phenotypes. Like body mass index may not be a disease; it's just a phenotype, or the height, or you know, eye color, or you know, whatever, hair color, and then they can do associations. A lot of these traits might be related, you see differences in genome now, and people are trying to understand, you know, whether a particular SNP or even a rare variant is associated with some trait. So based on the GWAS studies, there is some tools developed by the ENCODE consortium. And so on the class website, we have a tutorial to teach people how to use this. It's generated by also the ENCODE consortium. Basically, if you finish a GWAS study, you have, say, 50 hits from a GWAS, you know, you did SNP profiling of 100,000 individual, you identify like 50 SNPs. You want to ask what is going on with those 50 SNPs. So there are 2 web servers. You just input those SNP locations or the SNP ID. It can tell you which transcription factor might be binding to those SNPs or the nearby SNPs in LD. And you know, whether that could be related to the expression of some nearby genes. Okay, yeah, so in summary, so GWAS basically will just give you hits. They tell you some association, which is useful, but now people are asking why. And so association doesn't really mean causal. And so people are now looking into epigenetics to interpret and understand GWAS. And once they have a SNP linked to a DNA's hypersensitivity, very often the SNPs on the transcription factor binding side, on the DNA's hypersensitivity side, become the causal SNP, right? That's the real one that's, you know, like cause transcription factor to bind differently and then cause the nearby genes to be regulated differently. And so you can use you know, ChIP-seq or ATAC-seq or DNAs to identify regulatory SNPs that affect the transcription factor binding and influence the expression of important downstream disease genes. And then once you identify that SNP to be in the you know, transcription factor binding site, right now I would say getting the correct downstream target, it's still, you know, one at a time. People have to look, you know, really figure out which one is the target gene. They might use promoter enhancer interaction. And also if they know which transcription factor might bind there, they can use, you know, ChIP-seq data to help identify the downstream target gene and figure out which gene is the really important target. Okay, so these are all work that's really actively ongoing.